All right, so we left off looking at the various type of adrenergic receptors, and just to quickly refresh this, we talked about beta 1, beta 2, beta 3. Beta 1 and 3, we said, had equal affinities, whereas beta 2 had a preference for epinephrine over norepinephrine. And then we also spoke earlier about the adrenergic receptors as well, alpha 1 and alpha 2 subtypes of those receptors. So what we're going to do here is look at the mechanism. How do these receptors, once they bind these catecholamines, remember they can bind norepinephrine or epinephrine as a neurotransmitter or as a hormone. So when they bind this catecholamine, how do they actually go about bringing up a cellular response? So we're going to walk through the steps of that, and this is called the signal transduction cascade, okay, from the time of binding to the cellular response. So we want to think about alpha 1 receptors completely separately. They have an entirely different receptor makeup, they have a different amplifier enzyme, and they have different second messenger as well. So we'll speak about the mechanism involving alpha 1 receptors. Remember, these are usually found in the blood vessels, and they're somewhat more abundant than the other types of receptors. So let's look at how they bring about cellular response on their unique tissue locations. So first off, we want to think about let's think about this is a metabotropic receptor. So it's coupled to a G protein. Now the G protein here can be stimulatory or inhibitory. We're going to see some examples where this is an inhibitory G protein. But for alpha 1 here, it is a G stimulatory protein. That's what that subscript S stands for. So when this norepinephrine binds, the alpha subunit has a configurational change, and the subunit dissociates off and binds to the amplifier enzyme that is downstream in the membrane. In this case, it's phospholipase C. So the amplifier enzyme, we kind of talked about this loosely earlier, this is going to be responsible for making the second messenger. And the second messenger is going to ensure that it goes into the cell and does whatever is necessary to bring about the cellular response. Remember, this can be different for different types of tissues. It's going to be a different sequence of events on the lungs, on the heart, on blood vessels, etc. So the unique details we're not going to go into, but we'll talk about the amplifier enzyme and we'll talk about the different second messengers. So phospholipase C is the amplifier enzyme here. And again, the lipase tells us that this is going to cleave or cut something in order to generate its second messenger. So the cleaving of phosphatidylinositol bisphosphate 2 or PIP2 is what results in the generation of second messenger, which is IP3, as well as diacylglycerol. So IP3 goes into the cell. It helps to bring about a release of calcium from the endoplasmic reticulum inside the cell. And that flooding of calcium can bring about a cellular response, especially things like muscle contraction. When we get to that in the muscle chapter, we'll see how increasing calcium in the cytosol is going to be the way that we generate muscle contraction. So on an alpha 1 receptor, that flooding of calcium brings about the cellular response. The other second messenger is DAG, diacylglycerol. So both IP3 and DAG. DAG is going to be more involved in activating other enzymes in the cell, example, protein kinase C. And protein kinase is one of those modulating enzymes that can turn something on, turn something off. We talked about this when we looked at enzyme regulation earlier. And so protein kinase C is going to turn some other enzyme in the cell on or off, and that can bring about a cellular response as well. So both of these second messengers can help to generate the response on these receptors. Okay, so again, here that would be phosphorylating a protein, which can either turn it on or off, that allosteric phosphorylation, allosteric regulation that we discussed earlier. So that's the alpha 1 receptor, and it is unique in that it's the only one that uses PIP2. It's the only one that results in IP3 and DAG. Now, when you look at alpha 2 receptors and all the beta subtypes, so both alpha 2 and all beta receptors use the same amplifier enzyme and they use the same second messenger. However, their tissue location is going to be different, and what they do to that second messenger is also going to be different, okay, depending on the type of um, subunit that we see that it's coupled to. So for the alpha-2 receptors, we're going to talk about the pathway here, but notice it's the same enzyme, same second messenger, and so we'll talk about that for beta receptors as well. Now, to simplify this, I want to make this clear. For simplification, we're showing this on the same membrane. But remember, alpha-2 and the beta receptors, they're on different cell types, different types of tissue membranes. All right, but for simplicity, we'll discuss them together. So the alpha-2 receptor, remember, it's coupled to a G protein. This can be stimulatory or inhibitory. In this instance, it is a G inhibitory protein. So what we're going to see is that it's actually going to inhibit adenylyl cyclase production, which is going to inhibit cyclic AMP, its second messenger production. So whether the G protein is stimulatory with a subscript S or inhibitory with subscript I will determine the effect that it has on the amplifier enzyme and on the second messenger. So for alpha-2 here, it blocks adenylyl cyclase. When that subunit dissociates off, binds to adenylyl cyclase, it causes a blocking or inhibition, and that actually results in a decrease in its second messenger, okay? But we'll take a pause here, and we'll think about beta receptors as well. So for the beta receptor side of things, the subunit here, the G protein is stimulatory, and so the dissociation causes an activation of adenylyl cyclase, and then that causes an increase in cyclic AMP. Now, cyclic AMP is going to result in protein kinase, which again can phosphorylate some protein inside the cell and can turn it on or off and have that specific cellular response. Notice we're not going into the details of the cellular response because we're looking generally here at all beta receptors. So the alpha-2 blocks this pathway, and all beta receptors excite or activate this pathway, okay? Now here's a really nice condensed summary table to show you the receptor types, the tissue locations, where are they found, right? So alpha on our vascular smooth muscle, alpha-2 on CNS, it's on platelets, it's on some vascular smooth muscle, but again, alpha-1 is much more abundant. Beta-2 is going to be the heart mainly, but also kidneys and some parts of the CNS. Beta-2, sorry, that was beta-1. Beta-2 is going to be on the respiratory tract. We talked about the lungs and the effect it has on the lungs, the uterus, and then some very specific examples of blood vessels, like the ones that go to muscles, etc. And then beta-3 usually on adipose or fat tissue. We talked about their relative affinities. So alpha receptors prefer norepinephrine, beta-2 receptors prefer epinephrine, and then beta-1 and beta-3 have no preference. When we look at their signal transduction mechanism, what do they do to their second messenger and what is their second messenger? So alpha-1 second messenger is IP3, and it activates that because it is a G-stimulatory protein that's coupled to that receptor. Alpha-2 receptors are using cyclic AMP as their second messenger. Remember, adenylyl cyclase was their amplifier enzyme, but they inhibit that cyclic AMP production. Beta-1, 2, and 3 all are involving cyclic AMP as well, but they all activate cyclic AMP. And then their effects on the tissues. So I really want to point out here, this is an area where I usually see confusion, the effect that it has on its second messenger versus the effect that it has on the tissue. What's the ultimate cellular response going to look like? Is that going to be excitatory, such as contraction, or is that going to be inhibitory, such as dilation? So don't conflate these two responses. Example, beta-2 receptors we mentioned on the respiratory tract, they activate cyclic AMP, but that ends up being an inhibitory response when we look at the tissue level. Alternatively, alpha-2 receptors inhibit cyclic AMP, and that winds up being an excitatory response on these specific tissues. Example, contraction of vascular smooth muscle. So different, response happening at the cellular level versus what's happening on the tissue level. Okay. And then here are some drugs that actually have effects on the autonomic nervous system. And so this is how we can leverage our understanding of these receptors, of the adrenergic response, or the sympathetic response, as well as the parasympathetic response, to treat some common diseases and some common symptoms. For example, with asthmatic patients, they usually have airway inflammation, usually caused by, sometimes environmental um, allergens or environmental exposure, sometimes cold exposure, but that ends up constricting their airways and making it very difficult to breathe. So isoproterenol is a beta-2 agonist that can mimic the beta-2 response, mimic the sympathetic response, and bring about bronchodilation. So that can be very therapeutic for patients who have asthma. It's going to open up their airway and it's going to help to make breathing much easier. Similarly with pilocarpine. Pilocarpine is a muscarinic agonist. And so it's going to mimic the parasympathetic response. Remember, parasympathetic is rest, digest, which means we're going to have more glandular secretions. And so patients that have dry mouth or glaucoma, which is a decrease in the fluid of the eyes, we can have a mimic of the parasympathetic response to bring about increased oral secretion, increased salivary gland secretions, increased lacrimal gland secretions, which can correct or help reduce their symptoms. So these are just many of the drugs that have an effect on the autonomic system. Some are agonists that help to mimic that response. Some are antagonists that help to block that response. And so I don't want you to memorize this, but do understand sort of the practical and clinical application of these receptors.